by Dr. Madarcos to give you my lecture on macromolecules, particularly on amino acids and proteins. A general overview of uh, its general chemistry, structure, its digestion, and its absorption. Okay? Did Dr. Madarcos assign anyone for the invocation, a prayer, or before we start?
Hello, Doc. Ako po yung na-assign po.
All right. Please go ahead.
Start na po, Doc.
Yes, go ahead.
Good morning, everyone. Let's feel the presence of the Lord in the name of the Father, the Son, and the Holy Spirit. Heavenly Father, we thank you for this day and the new beginning of our class. Bless our minds with clarity, our hearts with patience, our hands with diligence. Guide our professor as he shares his wisdom. Help us understand our topic for today. In Jesus' mighty name, amen. In the name of the Father, the Son, and the Holy Spirit, amen.
Okay. Okay. So here's my screen. Okay. So if you have any questions, please feel free just to raise your hand in Zoom and then or send it in the chat box. And then you may raise your hand, and then once I call you, you may ask your question, or you may push it through the chat box. Okay. Hmm. Once again, good morning. I'm Dr. Aksai from uh, assigned by Dr. Madartos from the Department of Biochemistry. We're talking about amino acids, chemistry, absorption, and digestion. So we have 5 learning objectives for today. We first will define the basic concepts in proteins and amino acid chemistry, particularly for amino acids itself, peptides, and peptide bonds, and proteins. We can describe the chemical structure of an amino acid, and then classify amino acids according to their chemical structure of the side chain, their essentiality in nutrition, and their metabolic rate. Next, to discuss the 4 levels of protein organization in terms of structure and the bonds involved. And finally, to trace the digestion and absorption of amino acids from the diet. Okay, so let's start. Simple definition of terms muna. Amino acids. So they say it's an acid that contains an amino group. So these are organic compounds that contain both an amine group, hence amino, and a carboxylic group, which is a carboxylic acid, hence an acid. They are monomeric molecules that link to form chains of peptides and proteins. And biologically, in terms of biochemistry, only the L-alpha configuration of amino acids tend to form proteins. Okay. Also, yes, I will be giving this lecture in via PDF. Please send me the assigned person who I can send this to, class president or any deedles or class secretaries. Okay. Next, let's discuss the alpha carbon. The alpha carbon is the carbon attached that is bound to the carboxylic acid group. If you can see my pointer here. So this is the carboxylic acid group. Hence, this will be your alpha carbon. Okay. This is the carboxylic acid group. Hence, this is your alpha carbon. Any carbon From that point on, attached to the chain, based on the R side chain, is now your beta carbon. And if the side chain grows longer, hence it will become your delta carbon, gamma carbon, etc. Primarily for biochemistry, the more important part is we'll be talking about what is the alpha carbon. Okay, so all of the amino acids used in protein synthesis are based off alpha amino acids. There are some non-alpha amino acids present in the body as a result of metabolic reactions and intermediates, but those will be not be as focused in terms of basic biochemistry. You will encounter them in your physiology and possibly in your pharmacology. But in terms here, in the scope of biochemistry, we are focusing primarily on the alpha amino acids. Okay. Next, the chiral carbon. The chiral carbon is basically the atom that is bound to 4 different subunits. 19 of these 20 will contain a chiral carbon. So you might want to remember the exception, which of the ones later. You will see during the when we discuss the chemical structures. Next, what is a peptide? Peptides are linked chains of amino acid that are bound by peptide bonds. So these are longer chains of multiple amino acids. When you combine longer chains of peptides, you now call them polypeptides. Peptide bonds then are the covalent chemical bond linking a carbon from the carboxylic acid of one amino acid to the nitrogen of the amino group of another. So if this is one amino acid, you're looking at the carboxylic group, your C double bond OH, dropping the H that combines to the new a second amino acid through its amino group. The amino group is your NH2, dropping another H. So it's now C double bond O connecting to your NH. Okay. Next is a protein. So proteins then are complex macromolecules formed by single or multiple peptides formed from amino acids. This peptide superstructure determines its configuration based on its natural state, how a protein is found within the body, which then determines what it does and what it can do. So these proteins are then subject to physical and functional changes that can alter their actions and capabilities. We'll discuss that once we reach learning objective number 4. Okay. Any questions so far based on their definitions? Okay. If we have no questions, let's move on to our second learning objective. We discuss the structure of the amino acids from its components and conformations and its Zwitter ions. Okay. So here's the structure of an amino acid. It basically has 5 components. First, you have your alpha carbon connected to your the first carbon, again, connected to your carboxylic group. Then you have your amino group, your NH3 plus or your NH2. Your carboxyl group, your C double bond OH or C double bond O negative. A single hydrogen atom, and a side chain. All amino acids tend to follow this general structure. Okay? Next, for their stereochemical configuration. Enzymes that biosynthesize proteins can only work with L alpha amino acids. In terms of what we have in the body, again, only L alpha amino acids. When we mean by stereochemical configuration, it's based on how that amino acid is rotated in space. Um, how certain bonds affect each other based on their how they are attracted or repelled through their side chain components. This is basically deep physical chemistry, and we don't have to go through it. What I want you guys to remember is we're focusing on L alpha amino acids. Okay? Hmm. Next, side chains, or what is usually found in your books as R. These are the variable components of each amino acid. Um, what mostly you have to memorize in terms of its structure is, again, the amino group, the carboxyl group. It has one hydrogen atom all connected to their chiral carbon. The side chains are what makes each amino acid individual. There and it's from this side chain that will determine its function and its condition. Yeah. Okay. We also want to discuss the reactive subunits within the side chains. So these are non-hydrocarbon components or non-hydrogen, non-carbon components. They are reactive because they can be changed, they can be reduced, like the amino group. Your NH3 plus can be reduced to NH2. And your carboxylic groups, C double bond O minus, can be oxidized to COOH. Okay. Side chains with reactive units can also contain amides, hydroxyl, and sulfhydryl groups. And then your carboxylic groups, once again, can be oxidized. Zwitter ions, on this sense, are basically different forms of the same molecule, or tautomers, found in different states. So, this is a property of amino acids, particularly because it contains a group that can be potentially positively charged and potentially negatively charged. Okay, how is that? The charges are based on the different pH levels based on their pKa values. Please bear with me, this is a little tricky. These reactive subunits can be found via protonated and deprotonated forms. An example of a protonated form is your COOH that contains a hydrogen proton, and a deprotonated form is your COO negative, which is deprotonated. Okay, so let's use this for example of histidine. You can see it has a COO minus form, a COO minus in its carboxylic group, an H3 positive in its amine group, and its side chain contains more reactive subunits. Why does this happen? Just a quick context for acid-base chemistry. Amines, like your NH3, are weak bases. Carboxylic acids, or C double bond O negative, are weak acids. In the presence of a stronger base or a stronger acid, they tend to give up their um, they tend to either give up their hydrogen or be susceptible to more hydrogen. Example, your COO negative, your carboxylic acid, is a weak acid. In the presence of a strong base, a really strong base like sodium hydroxide, these will become very reactive. In solution, this strong base will dissociate. So you will have free hydroxide, OH negative, and your sodium will bond to water because it's in solution. What tends to happen is carboxylic acid uh, is less reactive to its hydrogen. Therefore, hydrogen tends to transfer to your hydroxide. Hence, this will when C double bond OH becomes C double bond O minus because hydroxide is very strong in attracting hydrogen. So again, since we added a strong base, you're expecting this pH to be higher. So the higher the pH, the less likely your C double bond O holds your hydrogen. In lower pH, carboxylic acid tends to hold your hydrogens more, and in reverse with your amines. Okay, so here's an example of Zwitter ions. Your four histidine Zwitter ions are located here in the bottom. Okay, below pH 1.8, you in this graph, or in your x-axis are your equivalents of hydroxide. Your y-axis is your pH. At the lowest point, one of the lowest points of pH at 2, or specifically at 1.8, histidine exists in this state. You have COOH, NH3 plus, and NH plus within your side chain. So in this point, you have a total electric charge of COOH is 0, NH3 plus is 1 plus, and NH plus is 2 plus. So below pH 1.8, you have a histidine of plus 2 charge. Understood? Please bear with me. Again, this is a tricky topic to understand. Please ask questions. Let me know if it's a little how I can help. Okay. Next, once we increase the pH, or we're in by increasing um, your hydroxide in the solution, at between pH 1.8 and 6.0, the hydroxide in the solution, like I showed earlier, tends to steal the hydrogen of your C double bond O, or your carboxylic group. You will now form this form of histidine, C double bond O negative, carboxylic group, NH3 plus amino group, sorry, and a side chain containing an NH plus. So -1 plus 1 plus 1, net charge is +1 histidine. Okay. Next, at pH 6.0 to 9.3, we're increasing the hydroxide in the solution again. OH has already stolen the hydrogen of C double bond O. It will also now take the hydrogen of your N side chain. Hence, your histidine at this point is net zero because it has negative 1, positive 1, 0. So net charge of your histidine is 0. And finally, above 9.3 in pH, your hydroxide has now stolen remaining the excess hydrogen of your NH3 plus, leading to a form of histidine that is C double bond O negative, NH2 0, and side chain 0. Why is this important? Because all of these forms are still histidine. but chemically they are in terms of chemical structure, they are different only with the presence of hydrogen and charges, and this is due to the pH that they exist in. Okay, so I would not expect to have uh, a plus one histidine when the pH is at 10, because past pH 9.3, all of the reactions taken place would have their hydrogen removed. Okay? Don't worry, I would not be asking too much on this on the exam side. I just want you to understand primarily that all of these forms of histidine is still histidine. Okay? It's just that sometimes the hydrogens are removed or present. Okay? And they're all still histidine. Yeah. Next. So these are the different forms and the states of amino acids that can be found. Also, side note, not every amino acid has side chains that have reactive subunits. I will explain. You will see in the chemical structures of the side chains. So now let's classify them. We have three forms of classification: the structure, the essentiality, and their metabolic fate. Okay. First, let's talk about your non-polar aliphatic amino acids. Non-polar because they are primarily made of hydrocarbons. Their side chain is all carbon and hydrogen. Aliphatic because they do not contain what we call a benzene ring, which is a six um, six carbon subunit that forms double bonds with one another. So these are all just long side chains of carbon, with the exception of glycine. Glycine's R group is a simple hydrogen atom. Okay, so going back, question: does glycine have an alpha carbon? You may answer via microphone.
Yes.
Okay, glycine has an alpha carbon. Question: does glycine have a chiral carbon?
Yes.
Okay, okay. Let's go back to the definition. An alpha carbon is a carbon that is connected. An alpha carbon is a carbon that is connected to a carboxylic acid group. So yes, you can see here that glycine has a chiral carbon, an alpha carbon. The definition of a chiral carbon is it has four different subunits. So glycine has a carboxylic group, an amino group, an a hydrogen atom, and another hydrogen atom. So there are two hydrogen atoms. Therefore, glycine is the single exception of amino acids that contain chiral carbons. Glycine is the simplest. It contains simply another hydrogen. So glycine, it does not have a chiral carbon, but all 20 have alpha carbons. Okay, but that is understood. Alanine is the next one, replacing the hydrogen with a simple CH3. It adds a methyl group. Okay. Next, proline. Okay. Some sources say that cysteine does not have a chiral carbon. Let's work on that when we get to the when we get to cysteine. Okay. We have a special case here in proline, because proline's R side chain also bonds to its amino group. In this case, proline is not an amino acid. It is considered an amino acid because it doesn't have an amine group. It has an imine group. Although we still classify it alongside its other amino acids. But strictly speaking, proline is called an amino acid. So its R side group is a methyl ethyl. It's a three carbon chain with the tail of that carbon chain attaching to your amino group. Okay? Yep, methyl ethyl propyl. Okay, sige. Next, your branched chain amino acids. Valine contains a branching CH connected to two methyl groups. Your leucine contains CH2 and then your branching CH3 CH3 group. While your isoleucine contains a CH, CH3, CH2, CH3. You see that they branch out. um, you have your branches within your hydrocarbon chain. These are your highly these are among your highly polar, non-polar amino acids. Okay? In terms of the exam, we don't expect you to memorize all the structures. What we just want you to do is familiarize yourselves with how these chains are made, how these chains look like, and particularly focusing on the exceptions. You have two exceptions here on this slide already. Glycine does not have a chiral carbon, and proline is not strictly an amino acid. It is an amino acid. Okay, next slide. You have your sulfur containing amino acids. Okay. So now we have your methionine in your cysteine. So there was a question earlier that cysteine does not have a chiral carbon. In terms of chirality, cysteine has your alpha carbon, your your carboxylic acid group, your amine group, a hydrogen atom, and a thioester bond. Um, a thiol group, which is your CH2 sulfur H. So as you can see here, the alpha carbon contains four different subgroups. So this should be still a chiral carbon. Okay. Methionine, on the other hand, has a S-methyl thioester. Thioether. Sorry. These will be notable, particularly with cysteine, in forming what we call disulfide bonds, where two cysteine units close to each other will tend to lose your your H of your uh, sulfidyl group that will now bond to another cysteine sulfidyl group, forming cysteine SS cysteine. Basically, both the sulfurs tend to stick to one another, kicking out their hydrogen. Okay. Next, the aromatics. Still non-polar. But here's what we see when we have a benzene or an aromatic ring. When you see this hexagon with a circle inside, each point is of that circle is basically see a carbon group that is filled with hydrogen, forming three double bonds in rotating form. The two double bonds, there are no two adjacent double bonds, but they tend to rotate where they can rotate within the ring. So if an example, if this is carbon, this point is carbon, it can be carbon, double bond carbon, single bond carbon, double bond carbon, single bond carbon, double bond carbon, single bond. Okay. So among these amino acids, they are the most water insoluble and they're the most hydrophobic. Basically, the more hydrocarbons found within the side chain of your the more hydrocarbons found within the side chain, the more hydrophobic it is. There are though side chains that are aromatic that contain polar ends. In terms of tyrosine, you have your phenol ring or an OH found within your benzene. So this OH is a very polar part of the subunit. Your tryptophan has an indole ring that contains ano to? That contains your nitrogen bound to a hydrogen. These make them more polar than your phenylalanine. Next, we move on to our polar amino acids. So these contains your reactive subunits. As an example, we have amide groups with your asparagine and your glutamine. Amide groups are NH2, or in this form, C double bond O NH2. You have your hydroxyl groups in your serine and threonine, which are your OH subgroups. Okay. Now we have um, polar amino acids that contain charges. All previous amino acids do not contain charges. So at neutral pH 7, we'll expect to have arginine, lysine, and histidine to contain a positive charge. For arginine, you have your guanidinium subgroup. Lysine will have an amino subgroup. And your histidine, as we discussed earlier, has an imidazole ring. All of the present hydrogens that can contain a charge can be reduced in the presence of a strong acid. Strong acid. Next, we have your negatively charged polar amino acids. So this will, at neutral pH, will have negative charges, each containing side chains based off carboxylic acids. Aspartate would have your ethanoic acid, ethyl group that has a carboxylic side, and propanoic acid or a propyl group with a carboxylic endpoint. This is your aspartate and your glutamate. Basically, this table will summarize everything that has been said. You have your forms of amino acids that can be either polar or non-polar. Whether they are, for those who are polar, they can be either acidic or basic or neutral. You have your aliphatic nonpolars, your aromatic nonpolars, and your neutral polars. Okay. If you can see here also, amino acids can be identified by, of course, their full name, a three-letter abbreviation, and a one-letter abbreviation. Not a lot of doctors tend to use the one letter. And in biochemistry, we're not really requiring everyone to memorize the one letter. We don't use it either. Uh, it- Although we encourage you guys to memorize them in their three-letter abbreviation. Okay. And finally, in the last two classifications of amino acids, their nutritional essentiality and their metabolic fate. When you digest when you intake your amino acids found in proteins, some of them can be synthesized from other amino acids that you take from the body. So these are the non-essential amino acids, because if you intake the essential amino acids, these non-essentials can be formed from the uh, from the amino skeletons of your essentials. So what are the essential amino acids? It will be this one: arginine, histidine, methionine, valine, isoleucine, phenylalanine, tryptophan, threonine, lysine, leucine. There is a acronym here called private Tim Hall that can be used to facilitate the memorization of your amino acids. So uh, PVT TIMHALL. I would recommend you memorizing which of the amino acids are essential. Okay. Next, once you intake these amino acids, if the amino acids are not used for building proteins but are used into formation of energy, you have three classifications. You have glucogenic, both glucogenic and ketogenic. Okay. So your glucogenic amino acids, once broken down, enter into your Krebs cycle and then form out to become components that can enter gluconeogenesis. Hence, they are glucogenic. For both glucogenic and ketogenic amino acids, tyrosine, isoleucine, phenylalanine, tryptophan, threonine, lysine, they can enter into gluconeogenesis or through ketosis that form ketone bodies. Finally, leucine cannot enter back into gluconeogenesis and only into ketone body formation, sorry. So what is good to memorize is the exceptions to the rule. Best if you understand that which are ketogenic and which are both, then the rest would be simply glucogenic. Okay. Okay. For leucine, it's based on its structure that it cannot uh the breakdown of leucine is not able to fully enter into the Krebs cycle to return back into to enter gluconeogenesis. So it can only its structure does not allow it to properly return back to become components for gluconeogenesis. Excuse me. Mhm. Okay. Any questions based on structure and on classification of amino acids before we move on? If no questions, let's move on to the levels of protein structure. So we've already talked about amino acids, and we talked about peptides, the peptide bonds binding them. Now imagine that we have full strands of amino acids in the tens to the hundreds, forming your polypeptides. Each of those strands each of those proteins, each of those amino acids affect the configuration of one another. You will rarely, if ever, find them in straight lines. So they will all fold and twist and turn to form clumps of peptides that will now form proteins. We describe those that structure in four levels. So in terms of protein structure, these are the four levels: primary, secondary, tertiary, and quaternary. So for primary, let's focus first. It's simply the linear sequence of amino acids in a polypeptide. Okay, not much to say, but it is the most immovable part of a protein structure that contains an unbroken chain. So how alanine is bound to glycine that is bound to serine, that is your primary structure. If you can lay all your amino acids in a protein out in a long line, it still holds its primary structure. Okay. Next, we focus on the secondary. Secondary is basically, if amino acids is basically your individuals in a line, your secondary is how those how people interact within that group. So here your short segments of polypeptides form repeating regions. Usually this is the effect of hydrogen bonds that form within the backbone carboxyl and the amino groups. They tend to form two types of configurations: alpha helixes and beta pleated sheets. Your alpha helixes happen because they rotate among a center axis, while your beta pleated sheets is basically like uh, a sheet of paper being folded upon itself. Segments that are formed that do not are not found within either an alpha helix or a beta pleated sheet usually form loops and or bends, which also serve their other roles. Okay. Next, for tertiary, this is now your three-dimensional configuration of your complete polypeptide, of a single complete polypeptide. So all the helices, all the sheets, the turns, the loops, the bends, of one single polypeptide form the tertiary structure, which can now have uh, which now form functional domains within the polypeptide. So each domain tends to have a specific function within the entire protein. The interactions you will find within the tertiary structures now include your salt bridges, hydrogen bonds, disulfide bonds, hydrophobic interactions. So these contain all total interactions of all side chains. The secondary is primarily based on hydrogen bonds alone. And finally, for quaternary, this is association of multiple polypeptides that now form, with all their twists, their turns, their sheets, and their helices, to form a complete protein. How this now looks in 3D is imagine multiple clumps of long strands bended, twisted together, now pushed together. And that each location of each polypeptide will serve their function. Yeah. Okay. Um, other lecturers may dive deeper on this, particularly for those who may be teaching on hemoglobin. Is a good example of showing subunits and um, how the structures will take effect with terms of their function. Okay. Last, we go to digestion and absorption of your proteins and amino acids. So here we trace the digestion and absorption from the diet into the bloodstream. So of course, for intake, we basically obtain them from dietary protein found in ingested foods. You have your plant, animal, and fungal sources of protein. Um, and each of those contain amino acids that can uh, be broken down into and absorbed into the bloodstream. Okay. First, indigestion. In the stomach and intestines, there are proteins that are designed, proteins that are broken down by very specific enzymes called proteolytic enzymes or proteases. They tend to exist in inactive forms known as zymogens. First and foremost, you will see in the these are examples, sorry. Um, to the left are their zymogens or their inactive forms. On the right is their active forms, and on top of the arrow is usually either their catalyst or the condition that they are activated. So here we see pepsinogen located in the stomach. It's activated in the presence of hydrogen ions or in an acidic medium. So this is a stomach enzyme where pepsinogen in the presence of their acidic medium in the stomach turns into pepsin. Trypsinogen in the presence of your enteropeptidase is activated into trypsin. Trypsin is important because it is a downstream reaction that activates all the other zymogens, your chymotrypsin, your proelastase, and your procarbopeptidase. Those activated by trypsin into chymotrypsin, elastase, and carboxypeptidase, respectively. If pepsinogen is a stomach enzyme, trypsin, chymotrypsin, elastase, and carboxypeptidase are found in the intestines secreted by the pancreas. Okay. So first, in the stomach, pepsinogen is activated through the acidic medium of the stomach, turning it into pepsin. Pepsin hydrolyzes peptide bonds of long proteins short to form shorter polypeptides. It's usually pepsin is more not wholly specific, but it targets its aim is to break down long proteins. But once you reach the intestines, pancreatic enzymes, trypsin, chymotrypsin, elastase, and carboxypeptidase cleave your polypeptides into amino acids or into shorter oligopeptides. Oligopeptides meaning short chain is think around 10 to 20 or even smaller amino acids in chain. Each of these, trypsin, chymotrypsin, elastase, and carboxypeptidase, are highly specific. They will tend to act on the peptide bonds of very specific amino acids. Which specific amino acids I can include in a table uh, with the handouts of this lecture. Okay. Next, intestinal epithelial cells can also produce aminopeptidases. So these are inside the cell. Once they take uh, short chain amino acids around the two to four, and intracellularly break down these short peptides into amino acids. Okay. Finally, for absorption, the amino acid uptake is using active transport via sodium-dependent transporters. In a short fish overview, when you have a low gradient inside the cell, it tends to absorb substances from an area of higher gradient. Inside the cell, you have what is called NAK-ATPase, an enzyme that releases sodium from inside the cell going out in exchange of taking in potassium. So you throw out sodium and you put in potassium. You use an ATP to do so. That I'm sure Dr. Madarcos will discuss in bioenergetics. So what's important is there inside the cell there is low sodium because of the action of NAK-ATPase. Because there is low sodium in the intestinal cell, what we call in the lumen side, the side facing the inside of your intestine, you have what we call sodium dependent transporters. So these transporters intake sodium, and through the action of intaking sodium, bring in amino acids. The sodium that is taken in is now flushed away by NAK-ATPase. But the amino acid now enters the cell. So now that the amino acid is in the cell, you'll see that it is technically linked to your sodium. Okay. Now that the amino acid is inside the cell, there are now facilitated transporters on the serosal side of the cell. Serosa meaning it's facing away from the lumen or away from the inside. Intracellular amino acids will now be transported into the blood via facilitated transporters. Again, on the serosal side of the cell. Okay. So again, most important parts to hear. Absorption is done via sodium-dependent transporters, and that is possible through a sodium gradient maintained by NAK ATPase. Finally, intracellular amino acids, once inside, are now used are now transported via facilitated transporters into the portal view. Okay. So short summary. Why are the structures? Why is all this important? Because each amino acid in the chain influences its neighbors. The presence of their side chains and the hydrogen bonds of their terminals folds to form a secondary structure, where in that point, the secondary helices, the sheets, the loops, the turns, the bends, can will affect the final shape of the single polypeptide. And finally, multiple polypeptides in combination affect and influence each other in a spatial arrangement to form a functional protein. This is where structure determines your configuration. Finally, the configuration will determine your function. So these domains formed in the secondary structures will serve specific roles. They can form channels, they can form receptors, they can form binding sites. Your alpha helices are looped around a space. That space within alpha helices can are examples of channels that can allow um, that can allow certain molecules to enter or not enter. The sheets can form sites that can become receptors or binding sites. The twists and turns can form epitopes. Hmm. So all these collective regions of your polypeptides act in tandem to form a dynamic and multifunctional protein. This is important because when you disrupt the protein through denaturation, through lysis, or via mutation, there is induced a loss of the function of that protein. Okay, that's it. These are my references. Hope you were able to take home from the lecture. Focus on what I tend to repeat, what I tend to highlight, because alam niyo na. Okay, please, if you have any questions,